Hello, my name is Hanin Elfadil, and this project is going to be about determining the response of the bacteria Pseudomonas aeruginosa to the polycyclic aromatic hydrocarbons naphthalene and phenanthrene, and whether it is able to degrade these two hydrocarbons. Polycyclic aromatic hydrocarbons, or PAHs, are environmental contaminants known for their toxicity and persistence in soil and water. This research aims to investigate both the bacterial growth and underlying genetic responses of the bacteria P. aeruginosa when exposed to these compounds. We hypothesized that the pH exposure would trigger the upregulation of catabolic genes, and namely NAHAC and CAT-A, and would lead to measurable degradation. Our objectives for this experiment were to optimize growth media for P. aeruginosa and find the balance between supporting growth and avoiding nutrient masking of the pH's effect. And our second objective was to monitor bacterial growth in 3 different treatments at 3 different concentrations. And our third objective was to quantify pH degradation using UPLC. And for our fourth objective, we needed to evaluate gene presence and expression using genomic PCR and qPCR. For our experimental setup, we had cultures exposed to 10, 50, and 100 micrograms per ml of naphthalene, phenanthrene, or a mixture of both compounds. We also had a bacterial control and an acetone control, since our hydrocarbons were dissolved in acetone. Optical density readings were taken at time intervals of 24 hours, 72 hours, and 7 days. The statistical significance was also assessed using a two-way ANOVA and a Dunnett's post hoc test. As you can see in figure 1, no significant inhibition of growth by PAHs was observed at the 24-hour time point compared to the bacterial control at that time point, while at the 72-hour mark, 10 micrograms per ml of naphthalene was shown to be significantly increased compared to the bacterial control at that time point. At the 7-day mark, the 10 micrograms per ml of the mixture of both naphthalene and phenanthrene was shown to also be significantly increased compared to the bacterial control. This indicates co-metabolism, where naphthalene can aid in the degradation of phenanthrene. Genomic DNA and RNA were then extracted after the 7-day period for analyses. Genomic PCR confirmed the presence of the NHAC gene and the CAT-A genes, as you can see in figure 2. qPCR only revealed the low-level CAT-A expression, where the treatments were not significantly different from control. The NHAC gene was completely undetectable. UPLC failed to detect any residual naphthalene or phenanthrene in our bacterial cultures, and that is likely due to low solubility, volatilization, or adsorption losses. The lack of detectable pHs in our UPLC analysis highlights the challenges in the bioavailability in aqueous media of these polycyclic aromatic hydrocarbons. The gene presence did not show a strong transcriptional response, suggesting that induction may require improved delivery methods or stronger inducers. Ultimately, P. aeruginosa harbors the catabolic genes that are responsible for the degradation of these PAHs, but it only shows in minimal transcription levels and no quantifiable degradation under the conditions that we use in this experiment. Future work should focus on improving PAH solubility. For this experiment, we use acetone to dissolve, which was toxic, but we had a control for it. So, including a positive gene expression control would also be good, and tracking intermediate metabolites over extended time courses to bridge the gap between the genetic potential and the functional biodegradation in real world scenarios. Thank you.